In this section, I'm going to talk about the prokaryotic cell envelope. The cell envelope is talking about the boundary of the cell, and in prokaryotes, it consists of a cell wall and a cell membrane, and then some bacteria also have an outer membrane in addition to that cell membrane. So there's a difference between the cell membrane and the cell wall. The cell membrane is that typical phospholipid bilayer that we discussed previously. The cell wall is a more rigid structure that provides cell shape and uniformity of the composition of the outside of the cell. So inside that cell wall, we have very specific molecules that usually repeat. In the case of bacterial cell walls, it's typically going to be peptidoglycan. In the case of other types of, like if we talk about eukaryotic cell walls, we have algae that has pectin, or we have fungi that has chitin in their cell walls. Other organisms don't have cell walls at all. Our cells do not have cell walls. The cell walls of protozoa don't exist, so protozoans do not have cell walls. So certain organisms have this cell wall, others don't. We as people, our cells do not have a cell wall, we just have that cell membrane. The cell wall and cell membrane act as a single protective unit, and they surround the cell, and they play major roles in what can get into and out of the cell, provide structural support and shape of the cell. The Gram stain is an extremely important staining procedure that allows us to differentiate between gram-positive and gram-negative bacteria. It's extremely important to know whether a patient is coming in with a gram-positive infection or a gram-negative infection, because the type of classification there determines what types of antibiotics we're going to be able to use, or what type of antibiotics are going to be effective against that pathogen. Now there's resistance that develops, and that's a whole separate topic, but fundamentally some antibiotics are designed to target specifically gram-positives or to target specifically gram-negatives. Or some antibiotics work better in gram-positives than gram-negatives and vice versa. So knowing this gram result and being able to provide this initial classification of bacteria is essential for healthcare, for treating patients, diagnosing these infections, and selecting the appropriate antibiotic. So because this is so important, you really need to know all of the intimate details about gram-positive, gram-negative, what makes them gram-positive and negative, and the whole staining procedure, which we will be performing in lab. So gram-positive have a thick cell wall, which has peptidoglycan in it, and then it has an inner membrane. Gram-negatives have an outer membrane, a thin cell wall, and then an inner cytoplasmic membrane. So this is a picture of a gram-positive cell wall. So you can see over here we have that thick layer of peptidoglycan, and then we have the cell membrane. That cell membrane is very similar in structure to the cell membrane that you'd find in our cells. But this cell wall is very thick and rigid and has that thick layer of peptidoglycan. Additionally, you're going to find some teichoic acids or lipoteichoic acids, and these are characteristic of gram-positive bacteria. Functions of these teichoic acids would be cell wall maintenance, enlargement during cell division, and it provides an acidic charge on the cell surface. Gram-negative bacteria have an outer membrane, a thin layer of peptidoglycan, and then an inner membrane. And the outer membrane is unique because the phospholipid bilayer is a little bit different. Instead of having the typical phospholipids, we have lipopolysaccharides on the surface of the outer membrane. These lipopolysaccharides act as a type of endotoxin, so that's something that can make us sick. When we start to attack, or when our immune system starts to attack these invading gram-negative organisms, the more they attack the organism, the more that they're going to release these lipopolysaccharides. And lipopolysaccharides then can have major effects on the body. They can cause a fever, they can cause delirium, especially in elderly patients. So that's a direct toxin where our immune system, the more we attack it, the more it's released and the worse the infection gets. So typically in those types of infections, the patient gets much worse before they start to get better. So since it's not spelt out very clearly and it's just a tiny little word over here, I'm going to write this out for you, lipopolysaccharide, because it's very important when we talk about gram-negative bacteria. And that is abbreviated LPS for lipopolysaccharide. And here it is. So lipopolysaccharide polysaccharide contains function as cell markers and receptors, and then an endotoxin that stimulates fever and shock reactions. Lipoproteins anchor the outer membrane to peptidoglycan, and then there's porin proteins that completely span the outer membrane. They only allow relatively small molecules to penetrate. The size can be altered to block the entrance of harmful chemicals, such as antibiotics, and they act as a defense against certain antibiotics. The outer membrane in gram-negative bacteria makes them resistant to some antimicrobial chemicals. They're more difficult to inhibit or kill than gram-positive bacteria. We'll talk more about this when we get to antibiotics, but if you're going to design an antibiotic that's going to kill bacteria, you want to make sure that it doesn't kill your own cells. So you have to find a target on the bacterial cell that the antibiotic can selectively attack without harming our cells. Now, peptidoglycan is a good target because we don't have peptidoglycan in our cells at all. Now, with those gram-positive bacteria, they have their cell membrane, and then they have that thick layer of peptidoglycan. So right away, that antibiotic can target that thick layer of peptidoglycan on their surface. With gram-negative, they have this outer membrane, then they have a thin layer of peptidoglycan, and then they have an inner membrane. It's difficult for antibiotics to be able to get into that outer membrane. So the fact that that outer membrane's there provides some resistance. Now, if the antibiotic does get through and does happen to attack the peptidoglycan is such a small component of the cell wall, it doesn't have a major effect on this organism. So gram-negatives are much more difficult to treat just because of the fact that they have this extra protective layer on the outside, this outer membrane. So now that you know the difference between gram-positive and gram-negative cell walls, I'm going to talk about the gram staining procedure. So in the gram stain, we would heat fix some bacterial cells to a slide. So typically what we do is we put a drop of water on the slide, then we aseptically transfer some bacteria to the slide, and we allow it to dry. Once it's completely dry, we're going to pass it through the flame 3 times. If there's no open flame, sometimes we'll allow it to sit on a slide dryer, and those can be attached to the top of an infrared incinerator, or they can be separate. And when they put the slide on top of there, that will dry it and fix the cells to the slide. Then we apply our first stain, and typically we're going to do this over a staining bucket. So we apply the stain directly to the surface of the slide and on the cells. When we apply the stain, the first stain is going to be crystal violet. And here you can see the crystal violet being added to the cells, and here you can see the crystal violet going into the membrane. Crystal violet is going to stain the outer surface of the bacterial cell. So in the case of gram-positives, it's going to stain predominantly this peptidoglycan layer. In the case of gram-negative cells, it's going to stain the outer membrane a little bit, and then it's going to really bind to this peptidoglycan layer that's underneath the outer membrane. Then we're going to add grams iodine. Iodine acts as a mordant. A mordant allows the primary stain to stick. So crystal violet is our primary stain, iodine is the mordant, and this allows that crystal violet to form a more tight complex with the membrane. So here you can see the crystal violet is just more deeply attached to the peptidoglycan, and the crystal violet is more deeply attached to the peptidoglycan here. Now here's the step that's the differentiating step. When we add the decolorizer. A decolorizer can be alcohol. Some labs use acetone, which is just nail polish remover. If it's alcohol, it's going to be a high percentage alcohol, like 95% ethanol. When we add the decolorizer, we pour drops of it over the slide until the solution runs clear. So we're going to drop, drop, drop. As soon as we see that it runs clear, we stop. What happens during this step is the decolorizer is going to try to wash away some of the purple from this peptidoglycan here, but it's not successful because this layer is so thick. It maintains that peptidoglycan or that crystal violet stain. Now in the case of gram negatives, when we have that outer membrane and then that thin layer peptidoglycan, this decolorizer is able to completely wash away everything here. All the purple goes away, leaving the cells clear. Because these are clear now, when we go to the next step, we can see that when we add safranin, which is the counter stain, adding this pink safranin color to a purple cell is just going to make it a more pretty purple color. But when we add pink safranin to the clear cell, it turns it pink. So at the end of the staining procedure, gram-positive cells are going to appear purple, while gram-negative cells appear pink or red in color. So some terms to make note of: the crystal violet is the primary stain. The Gram's iodine is the mordant. The alcohol is the decolorizer. And the safranin is the counter stain. So again, the differentiating step is this decolorizer step. The reason why the gram negatives become clear is because they have such a thin layer of peptidoglycan, it can't hold the stain. When the decolorizer is added, it dissociates those stain and it turns the cells clear. There are non-typical cell walls. So non-typical cell walls lack cell wall structure of gram-positive or gram-negative bacteria. The cells of Mycobacterium and Nocardia are composed of unique lipids. Mycolic acid or cord factor, they're very long-chain fatty acids, and they contribute to the pathogenicity of these organisms. They make them resistant to certain chemicals and dyes, and they require that acid-fast stain in order to diagnose. So if we look at tuberculosis and leprosy, we need to use that acid-fast stain in order to visualize those organisms. Mycoplasmas do not have a cell wall. Their membrane is stabilized by sterols and it's resistant to lysis. Mycoplasmas are extremely dangerous when they're found inside of a patient because they're nearly impossible to get rid of. You can't kill them with typical antibiotics, and they're a pretty hardy microorganism once established. Mycoplasmas usually only affect the immunocompromised, so they're opportunistic pathogens. But they have been known to cause a few infections in healthy people as well. The cell membrane structure is that phospholipid bilayer, which we discussed previously. So the lipid bilayer is embedded with proteins. There's phospholipids and there's proteins. Mycoplasmas contain a high amount of sterols in their cell membrane, and archaea contain unique branched hydrocarbons rather than fatty acids. The cell membrane functions in energy reactions, nutrient processing, and regulating the transport of nutrients and wastes into and out of the cell. Energy reactions. So if you recall, in a eukaryotic cell has a nucleus, the endoplasmic reticulum, Golgi apparatus, and then they have mitochondria inside of them. And mitochondria are the energy centers of the cell. If you recall a prokaryotic cell has some ribosomes, some genetic material that's coiled up in the inside, and that's basically it. Now there's a cytoskeleton and there's some inclusion bodies that can form and so forth, but there's definitely no mitochondria. So all of the reactions that would happen in the mitochondria typically happen across this cell membrane here. So that's one of the most important functions of the cell membrane for prokaryotic cells, is because they don't have a mitochondria, they're going to have all of those reactions happen on their cell membrane. The cell membrane is selectively permeable, so it has selective permeability. Water and small uncharged molecules can diffuse freely. Special carrier molecules or mechanisms exist for the passage of most molecules. And then secretion, that's the discharge of metabolic products into the extracellular environment. So those are all part of the functions of the cytoplasmic membrane.